Hi, Michelle Glass here. Welcome back to our second part of chapter 22. So we are still looking at the lymphatic system, which is where we left off. We kind of stopped in the middle of that chapter with the discussion of the non-specific defenses, and we're ready to finish chapter 22 by exploring those specific defenses. Now, specific defenses can also be referred to as adaptive immunity because these are responses that require exposure to a pathogen before that defense mechanism is in place. That's different from the innate defenses that we talked about, or the non-specific defenses that we talked about before, which are defenses that you're automatically born with. You don't have to be exposed to pathogen in order to form skin. You're born with that skin. So that sort of idea. Okay, so let us get started with an introduction here. With specific defenses, this is going to involve our lymphocytes, specifically our T cells, which we see are described as being involved in the cell-mediated defense. We'll see that means like they specifically target and attack and kill infected cells. And then we have our B cells, which are described as being involved in antibody-mediated defense. Now notice I inserted my little plasma cell in between, and the reason here is I want you to be remembering that the B cell itself doesn't produce the antibody. The antibodies are coming from the plasma cells. So we get our B cell that's activated, it makes the plasma cell releases that antibody. So we'll see that in the future as we get into some more detailed descriptions. Okay, and before we can go further, I've used the word antigen. We need to make sure we're really clear on what this means. Antigens are typically protein markers on your cells. So remember we talked about glycoproteins as being like little name tags on our cell membranes. Well, they're not just on our cells. All cells have some protein markers on the outside. Even though I'm thinking about antigens primarily as proteins and talking about antigens primarily as proteins, they can be lipids or polysaccharides or even nucleic acids. So there is some variety there in the molecular structure. The key thing is that an antigen is going to be one of these markers on the cell that generates an immune response. So it's called an antigen because it triggers or generates the immune response. What we see happening is those lymphocytes have to actually see the antigen before they can be activated. So once the lymphocyte is in the presence of the antigen, then we're going to get that lymphocyte activated. When we talk about a lymphocyte being activated, what that means is it's going to do cell division. And what it's going to do is to make more of that type of lymphocyte. So it's making clones. And all of those clones that it's making is going to be sensitive to exactly that same antigen. So whatever antigen it was that triggered activation of the lymphocyte, that will be the antigen that all of these clones are sensitive to. When we're talking about specific defenses, when we're talking about T cells and B cells, it's like a lock and key model level specificity. It's like a puzzle piece level specificity. So it's not just like any old lymphocyte is going to be activated by any old antigen. No, it's a specific matchup. So our bodies have to be producing millions of different types of T cells and millions of different types of B cells. So no matter what antigen we're exposed to, we can have an immune response against that antigen. So that is part of our genetic makeup, our diversity of our lymphocytes that we are producing. Okay. When we talk about adaptive immunity, again, this is different from innate immunity because it requires exposure. So even though you have millions of these different lymphocytes as part of your genetic inheritance, they're not activated until they have first seen the antigen, which means exposure has to happen. Now we can have natural exposure, or we're going to look at, in a few minutes, the other side of that, which is an artificial exposure. If you are naturally exposed, let me rephrase, there are two natural ways of getting adaptive immunity. So we have what's called naturally acquired active immunity. Naturally acquired active immunity. This is where you get sick. That's how your lymphocytes are exposed to the antigen. So your lymphocytes are activated. They make those clones sensitive to that particular antigen, and they remember it. So the next time you see that antigen, you can attack fast and strong, and you don't even know you've been exposed. You don't get sick, or you don't get as sick on that second exposure. So the significance of the active immunity here is the formation of memory. There's another way to have antibodies. This is the naturally acquired passive immunity. Passive means you're not actually making those antibodies, which means you haven't actually activated any lymphocytes, which means you don't get any memory. So there's really two ways that this happens naturally. One is some antibodies will actually cross the placenta so that you can be born with some antibodies already in circulation of that infant. And then, of course, antibodies are crossing with the breast milk. So this is an important contribution that the mother can give to her infant, such that the infant already has antibodies in circulation, so that if your infant is exposed to an antigen, while the infant's body is doing the naturally acquired active immunity, you still have some antibodies on hand so that the infant doesn't get as sick or sick. When you get to passive immunity, you don't get any kind of memory as a result of that. But hopefully this is kind of buying you time while you do have your own response and you do produce that memory. On the opposite side of things, we can have artificially acquired immunity. So you can have artificially acquired active immunity. This is what we're most often familiar with. This is where you're receiving a vaccination that has pieces and parts of a pathogen. Basically, you're injecting the body with antigens such that your lymphocytes see it, your lymphocytes activate, they make clones, they have memory cells. But the benefit of this method is, of course, you don't have the risks that come with the actual illness. So you're not getting sick, but you're still having an immune response and you still develop that memory. And then you can also have artificially acquired passive immunity. And again, if it's passive, that means your body's not doing the immune response, you're not getting the memory, but you are, in this case, getting an injection of antibodies. And so this is allowing you to fight off that infection, without getting sick. Or we talked about the Rogaine shot as being important as a preventative for erythroblastosis fetalis. So we talked about that back in chapter 19. So that was an example of that. Okay, as we talk about adaptive immunity, we have 4 important characteristics that we need to know. So the first is that it's a very specific. So we've tried to sort of already begin looking at this idea. The molecular structure of the antigen is what's triggering activation. So that means the lymphocyte has to be able to actually see that antigen. So that means it has to be able to recognize and bind to the antigen. We talked about antigens as being proteins. You should already know that protein structure is highly complex, highly variable, and usually there's this idea of like the lock and key model or the puzzle piece model when we're talking about protein shape being important in protein function. So because your lymphocytes have to be able to see those antigens, then you have to have a lymphocyte that's able to see each of the different types of antigens you might be exposed to. Very specific. And our adaptive immunity is very versatile. So it's okay that it's super specific because we have, as part of our genetic inheritance, millions of different lymphocytes. So they all have their eyeballs, you know, not really, right? But they're all looking out for all the different types of antigens that they can possibly see, so that you should be able to mount an immune response no matter what antigen you're exposed to. And then the third important characteristic is the memory. So I referred to this already when we were looking at different types of immunity. Being able to actually do your immune response is important because you get the memory with that. The memory allows you to respond faster and stronger and have a more long-lasting counterattack with that second or third or fourth or fifth exposure. So this is the whole reason we do vaccinations. Vaccinations give you the memory, and you don't risk being sick in order to get that memory. And then our final and our fourth characteristic here is tolerance. So not only are those lymphocytes specific, and not only do we have millions of them so we can see all of our different possible antigens, and not only do we have the ability to remember those that we've seen before, but we also can recognize what antigens are foreign and what antigens are belonging to ourselves. So we have tolerance. So the lymphocytes recognize, oh, hey, dude, what's up? You know, you belong here. And then you don't belong here. Let's get you out of here, right? So we have this ability to see antigens as either self or as foreign, and then only activate, or usually only activate, in response to those specific foreign antigens that we're able to see. Okay, so that is it for now.